Hi everybody, my name's Jo Burgess, I work for IEL and my background is biochemistry and microbiology, so I'm going to do a quick Virology 101 on SARS-CoV-2 and the disease it causes, COVID-19. The words and terms that I'll be using and that you will have seen in the literature include COVID-19, which is the name of the infectious disease caused by the most recently discovered coronavirus. The name of that virus is SARS-CoV-2. SARS means severe acute respiratory syndrome. A virion is a complete viral particle, so this is the infective particle. It consists of the virus's genetic material, which is RNA, surrounded by a protein shell, and this is the infective form of the virus. If you don't detect a whole virion, you haven't detected an infective virus. Fomites are inanimate objects that become contaminated with infectious agents, virions, and they serve as a mechanism of transfer between hosts. The classic example of a fomite in this case is a handrail or a doorknob, where virions deposited by one person can be potentially transmitted to the other person who later touches the same object. Coronaviruses were first discovered in the 1930s in domestic chickens, and human coronaviruses were first discovered in the 1960s. SARS-CoV-2 is the 7th human coronavirus to be discovered. SARS-CoV-2 belongs to the species severe acute respiratory syndrome related coronavirus. It's in the genus beta coronavirus and the family Coronaviridae, which consists of a group of enveloped viruses with a single-stranded positive sense RNA genome. SARS-CoV and SARS-CoV-2 are also distantly related to Middle East respiratory syndrome coronavirus, or MERS-CoV, which belongs in the same genus, beta coronavirus. The mild illnesses that coronaviruses cause include somewhere between 15 and 30% of cases of the common cold, and then the more lethal varieties include SARS, Middle East respiratory syndrome or MERS, and COVID-19. The current classification of coronaviruses recognises 39 species that belong to the family Coronaviridae. This phylogenetic tree shows 5 species of severe acute respiratory syndrome related coronavirus of the genus beta coronavirus. The red text, SARS-CoV-2, SARS-CoV, for example, highlights zoonotic viruses, so those are viruses which can jump to humans, with varying pathogenicity in humans. The orange text further down highlights common respiratory viruses that circulate in humans, but don't make us very ill. You can see from the shape of the tree how closely related these viruses are, and this is good news. SARS-CoV-2 has a 78% nucleotide identity with the original SARS that caused an outbreak much earlier in this century. This is important because it means that what we already know about the way SARS-CoV behaves is likely to also be relevant to SARS-CoV-2 as well. All of the viruses in the order Nidiovirales are similar to this. They contain the virus's genetic material, its RNA, enclosed by a lipid bilayer. The membrane glycoprotein, M, the envelope glycoprotein, E, and the spike protein, S, are all anchored in that grey lipid bilayer, the circle in the diagram. And all of these structures together form the viral envelope. Inside the envelope there is the nuclear capsid, the blue squiggle, which is formed from multiple copies of a nuclear protein, or N, which are bound to a single-stranded RNA genome, which is a continuous sort of beads on a string type conformation in this case. SARS-CoV-2 has the largest strand of RNA yet discovered in a coronavirus. It's over 30 kilobase pairs. This is really good news because it makes DNA detection, RNA detection, even easier than with viruses that have smaller RNA strands. The lipid bilayer envelope, the membrane proteins, and the nuclear protein protect the virus when it's outside of a host cell, and the spike protein is what enables the virion to attach to and infect a host like you or me. All of the enveloped viruses are relatively fragile, and they can be deactivated by any kind of soap. The soap acts on the lipid bilayer, detaching the hydrophobic lipid from the rest of the envelope and exposing and destroying the inside of the virion. So this is the disgusting bit, coronaviruses in water. Both viable SARS-CoV-2 virions and viral RNA are shed in bodily excreta, including saliva, sputum and feces, and these are subsequently disposed of in wastewater. Human coronaviruses are known to replicate in the gastrointestinal tract and caused gastrointestinal symptoms such as diarrhea in about 10% of COVID-19 patients. The SARS-CoV RNA loads can be extremely high, both in feces and in urine, and all of this viral load enters the wastewater system. Positive isolation from feces has been found in over 20% of patients, even after they've apparently recovered from the disease itself. And clinical studies have also shown fecal shedding of SARS-CoV-2 RNA for 7 weeks after the symptoms, and in over 80% of people that were asymptomatic, so they had a negative throat swab result, which we take to mean that they don't have the disease, but the feces show that they do. We also find that between 17 and 30% of infected individuals with COVID-19 are completely asymptomatic, but we find RNA from the virus in their feces as well. So what does this mean? This means that although it's believed that the major transmission route of this virus is inhalation via person-to-person, aerosol or droplet transmission, and fomite-to-hand contamination, wastewater and receiving water bodies have a globally significant role as a potential source of epidemiological data and also have a role to play in public health risk. Our knowledge on the presence and persistence of coronaviruses in water is limited, probably due to the lack of previous environmental investigations focusing on coronaviruses. Enveloped viruses are thought to be primarily spread via person-to-person contact rather than through the fecal-oral route. That's been postulated but not yet proven, although there are data emerging on that topic, and their presence in wastewater requires some very careful interpretation. The presence of the coronavirus RNA in wastewater has not yet found widespread use as a disease surveillance tool, but this is gaining traction, and this nice big strand of RNA is relatively easy to detect. But finding the RNA in the wastewater or the surface water doesn't mean that the water is infectious or contains virions. The standard virus concentration methods used on other kinds of viruses don't work very well for enveloped viruses, particularly in water and on surfaces. So a new standard method needed to be devised and agreed upon. This graph shows you why coronaviruses are not considered to be waterborne diseases. This line here shows you the persistence of poliovirus in water, and you can see that poliovirus persists in water for over 20 days after the virus has been deliberately spiked into a water sample. This HCoV sample, this line here, shows you that the virus titer found in a sample drops off very quickly over a 10-day period. So at the moment we believe that waterborne transmission is not very likely for coronaviruses, but we don't know for sure yet.